Juan Bada presenting. He's currently a postdoc in Lausanne at the EPFL. And he's actually working on sensing single molecules through a biological nanopore. In this case, it's aerolysin. Yes. And it has this technique of having an artificial bilayer, introducing a nanopore, and then sensing molecules has become so sensitive that you can also sense post-translational modifications in peptides and proteins. So really looking forward to this talk. Works, no? All right.
Yeah.
There we go.
Cool. Well, thank you very much, Conrad, and thank you, Nanyan, for this invitation here to give this talk about. Here we're going to talk today about nanopores and how we can, let's say, detect, sense kind of PTMs in kind of some neurodegenerative protein. Okay, so I should say I'm part of University of Geneva and also from EPFL because Chan recently, my boss, she moved from EPFL to Geneva. That's I just want to make it clear here. So in this case, let's see, yes. So just to put everyone on the same floor here, I'm just gonna introduce first a little bit of nanopore, what it is, okay? So you imagine it's a kind of single molecule technique that is working at the nanometer scale, okay? And what you can sense is like different kind of analyte, from going from sugars, you can go PEG, you can go peptides, and you can go also DNA, okay? And what you have been, what I'm just showing here is like this, based on this Coulter device. Yeah. Okay, so here, just like demonstrate what it looks like a nanopore, I would say. So you have just a hole. Imagine you have like a flux of ion going through. And this would be at the bottom here, you would see like the kind of ion that you have. And something goes here inside, like the DNA, and will block like the ion or the flux of the ions going through. I just put the picture over there of the elephant going versus an ant. So the door would be your kind of pore. And then if you have an elephant going through, you would block more than just a small ant. And we use here like just filled with electrolytes. So we just have potassium, we can have sodium chloride, lithium chloride, like the nanopore field is a bit crazy. We have like different kind of, let's say, electrolyte. And the next one I just want to show is like what we use as kind of nanopore, okay? So we're not using really ion channels, we use something a little bit bigger. I will show you in the next slide. And just to keep like people with us, I would say like what we have is what we usually call the open pore current here, as you can see. And then what we want to see is like different analytes. So at the bottom here, A, B, and C, as you can see, like block for different time that we will call the dwell time, and also block differently if they are longer, bigger, or whatever. So they kind of have very significant signal. So as I can say here, what can we use as nanopore? And in fact, what we use as nanopore here is what we call the pore-forming toxin. It's a very interesting kind of proteins because they are soluble protein, which in contact with some enzyme suddenly have been triggered and enter into the membrane to form a pore, okay? So this is usually a defense mechanism of some bacteria that when they get attacked, they release this kind of soluble protein into the media, and then the other bacteria that is there is trying to digest this protein, and when digesting the protein, in fact, it kills himself, okay? So we use this kind of nicely, let's say, biological killer to have a nice pore that is reproducible. And in fact, what is really cool with this kind of pore-forming toxin is like they are not selective to kind of anything. So in fact, they translocate, as you can see here, molecules, protein, ion, sugars, okay? And of course, like also some ATP. So one of the pore-forming toxin we use in the lab is the aerolysin. I say here is the narrowest biological pore. I would say one of the narrowest biological pore because every day we discover more and more. But what you can see here is depicted is like how it looks like. So it's a kind of 10 nanometer long, very nice beta barrel protein, where you have like a very small kind of inner diameter, approximately of 1.8 nanometer. I just depicted on the bottom left what you have like some key amino acids that are very, let's say, selective here. So one for the pore forming, like really important, for the selectivity. So what we call really like where we're going to play a little bit with the sensing, if you want, and also some of the sensitivity. So here, I just put next to it like different mutants we have been trying, where we can see like we have like different mutants at these 2 points, like the R220 and the K238 that we have there. And this has been done by Chan, like previously, where she have been like looking into different mutants, where you can see how the diameter, in fact, of the inner pore is different. So this is what we call, in fact, the constriction, okay? So you can see, for example, here that the K238A, the K238Q, that maybe I can show here. Is it working? No. This one, no, well, sorry. The K238 and the K238Q, they are like very like, very narrow at this point, okay? So in fact, we were doing like different ones, and for the purpose of the sensing of this project, we use the K238A here, okay? And what we're gonna sense is in fact the alpha-synuclein, because the alpha-synuclein has been like largely like understood as one of the most degenerative peptides, or protein in this case. It's mainly usually found in the brain and in the neurons. And some of these PTMs that you can see there on the kind of bottom left, you can see that are triggering multiple effects, okay? And majorly what they lead is to neurodegeneration and Parkinson's disease. The problem of this is like when people usually start to detect them nowadays, it's a bit already too late to have some treatment. So what we are trying to do here is to try to have like a very early stage clinical kind of case where we can detect this PTM in order to try to find new cures and treatment for that. And the problem of this alpha-synuclein, where we have most of the PTMs, is what is called this acidic region C-terminus, is not very well adapted for mass spectrometry because so negatively charged that in fact takes so much charge that it goes directly to the noise of the mass spec. And effectively, if you start to add phosphorylation stuff, you add more and more charge, and in fact make it even more difficult to detect. So we have to find a new way to really detect all these peptides and these PTMs. And that's what we're doing here with the aerolysin nanopore. So we took a small portion, not the full C-terminus first. So this is like a synthetic peptide that we took where we have like some phosphorylation, some nitration, some oxidation. And what we're trying to see first is what kind of signal do we have when we have the nanopore? And you can see in 1 molar concentration here at pH 7.4, you can see like we have very nice kind of distinguishable events. So you can see this kind of negative spike that result in 2 kinds of population that is also depicted in the middle. And then when we put urea, you see that the signal is completely different. We just can kill this kind of 2 population and only get 1. And in fact, it's due to this kind of IMC, like you see at the very beginning. It's kind of a hydrophobic cluster that goes together. And I will show you in a minute why it's so important. So in fact, when this cluster there, this local conformation is still present that you cannot really disrupt with urea, in fact, you have 2 kind of level signal. And when it's disrupted, you only get 1 level signal. So in fact, already with this kind of experiment, we can already see that we can observe very local conformation of a peptide that we cannot see with some cyclodicresum, for example. So then what we did was just to try different PTM, okay? So this is all synthetic peptides, and we were just adding some more and more PTM. So first we have like let's say like mono PTM, so 1 PTM at certain point, and then we also play with different one when we have 2 PTM and 3 PTM on the same peptide. And you can see how the signal, especially here on the bottom right, how it's affected by the presence of this PTM. So you see either the signal becomes super long and 1 very nice blockade, or it becomes like 2 blockade. And again, when you go to this IMC kind of region, you can see that, for example, the PS129, you again got like a very kind of 1 single event. Basically there, because you are also disrupting this kind of hydrophobic kind of patch. The same happens for the NY125, it's the same principle. So really when you go there and you start to add some PTM, you affect the signal of this in the nanopore. And once we did this kind of library now that we have here, we were interested to see, well, what can we do? We need like some tool to really like distinguish that. We're not gonna check individually all these guys by hand to see, it looks like wild type, it looks like PS129 or whatever, right? So we need a kind of automatic tool to really assess who is who. And this is where we, in fact, come to the machine learning and even the deep learning here, where we use like this kind of new tool to really put some of the data we had there and just to assess who was who in kind of this kind of library, okay? So we did some mixture experiment that you can see on the second row here, where we were trying to put different like peptides together, so the wild type, the NY and the PY, for example, here, and we check experimentally what it looks like, and then we assess by machine learning, we asked machine learning to see what kind of ratio we could have observed there, and you can see that between the theoretical and the predicted one, you have a very good kind of ratio. So it's really predicting very well for at least a mixture experiment. And then we have what we call at the bottom here, on the bottom left, you have the confusion matrix. So it means how far is the model able to predict the actual peptide that is there. And you can see that, for example, the PS129 that I told you is the only validated biomarker for the alpha-synuclein. You have a 93% good accuracy that it's able to identify. You can see some of them are a little bit lower, but nevertheless, they're all like very nicely detected. So once we had that, this tool, in fact, it's something that we also put it in one of the previous paper when we're doing the polymer sequencing, like something similar to DNA. We also use the same kind of machinery to also assess who was who in this kind of thing, kind of signal. So once we had the tool to detect this, we went a little bit further and say, okay, now that we have the tool, we need to be sure that we can detect this on a kind of clinical level. So what we did was to check if in, for example, in red blood cell, where we have like a certain amount of alpha-synuclein, what was kind of the background that we have there, right? Because in Nanopore, as I told you, we can detect basically anything that would be there. So we need to be sure that when we're going to put some red blood cell, we're not going to have a million signal that is not whatever we want. So we need to also to kind of adjust and see what we can do. So here what we did was just to take some red blood cell from a collaborator, like Professor Hilal Lashwell at EPFL, and we tested like to see what kind of ratio we can use until we get some certain kind of signal there. And you can see that even like at 1-to-1 ratio of red blood cell compared to analyte or electrolyte, we didn't have that much kind of very long signal that looks like the alpha-synuclein. So we're kind of pretty surprised, I would say, but also happy that we're not having that much like signal. About this sample, I should say that we also removed the hemoglobin from here using a commercial kit just to be sure we were not also, because hemoglobin is very abundant there. And then on the right-hand side, what we did was just to take this kind of matrix of red blood cell and spike some of our peptides. That was the one I just showed you before, like just the wild type, and just to see if it was still being able to be detected. And you can see that we even see here even the 2-level nicely in this kind of thing, in this kind of signal that we have here. So in terms of red blood cell matrix, there was not really a kind of problem. The other problem, in fact, is that, as you maybe are not aware, but I will tell you now, the alpha-synuclein concentration in red blood cell is very low. It's about 39 nanomolar. So we need also a way to kind of improve the signal frequency, so how much sample can go through. And the way to do that, you can see like the signal frequency that we have for symmetric conditions, so basically the same amount of electrolyte on each side. When we have a certain amount here of alpha-synuclein, it's very low. So you have one signal every 2 seconds. So if you need 10,000 events, it will take you kind of a few hours to have the responsive what is there in the sample. So to increase this kind of event frequency, we use here what we call the asymmetric buffer condition. So we play with the buffer. You see that in one side we have very low salt, and then on the other side we have a very high salt. And what is basically is driving everything go through the nanopore. So the event frequency, in fact, is increasing a lot, and that's in fact lower our limit of detection. So we even went from something like nanomolar concentration up to 100 picomolar concentration. So it's really a nice way to kind of let's say, popping up the signal of the alpha-synuclein, but also of all the other stuff that is there, because again, it's non-selective, so you basically see a lot of stuff. But thanks to our machine learning, we can identify them back. And the last point here is like, because this is all very nice, we can see that, and this peptide is just chemically synthesized, okay? The way would be like, how can you do that clinically to have this kind of peptide? And that we have been looking into it is like, here over there, you have like the full sequence of the alpha-synuclein. But we found an enzyme, which is called the catepsin D, CTSD, that cut exactly this peptide. So in fact, you can basically take the alpha-synuclein enriched, for example, with some antibodies or something like that, treat it with your enzyme, the catepsin D, and then back identify again this peptide that we just have the library for. And in fact, we did that, and you can see that we have like several fragments there that we put it into the nanopore experiment. Again, now you have like multiple peaks because now it looks like a more real, let's say, environment because you have the full peak of alpha-synuclein. And we try to get it back to see if the deep learning was able to really assess again this guy there. And you see that we have very high accuracy, like saying that we are able to back identify this in a very different matrix, okay? So we first, we try with the full length alpha-synuclein, but we also try with a shorter length. Just first we did the shorter to be sure, to be honest, and then we went for the full protein just to be sure it was able to see. So in fact, it kind of opened a kind of way where we can imagine that everyone is using now ELISA kind of way to check if alpha-synuclein is there. So it would be also a kind of way where we can isolate this kind of alpha-synuclein first, and then we chop it with the enzyme, and then we put it to the nanopore. So that would be kind of what we aim to do here. And in fact, it's basically going to my conclusion here, like basically we have like detection of small peptides by the aerolysin where we can discriminate, in fact, several like post-translational modification. And we're already a kind of small library where we can think it's very interesting already as a starting point. We use the machine learning just to identify because we don't want someone to spend a day just to tell you, yes, you have PS129 because I checked 10,000 events and I think we have some. So that's why we need this kind of machine learning there. We have seen that the red blood cell here is not interfering with the detection of the alpha-synuclein. In fact, it's not also even interfering with the membrane, which is also something that is quite interesting because we try to do that with plasma, but plasma just break up the membrane. So interesting that the red blood cell is not apparently doing anything here. And we can also lower the limit of detection very low with this asymmetric condition. And finally, I think we can generate this peptide with enzyme. Again, biology is helping us here a lot, I would say. Thank you, the biology and millions of years of evolution. We just have to find the good enzyme for the good purpose. And we think this could be used like not only for the alpha-synuclein, where in fact we're also trying other neurodegenerative peptides that have the kind of same pattern in terms of PTM and stuff. And we think it could be a kind of pipeline we can implement to really have a look at all these kind of neurodegenerative peptides. And because it's not, in fact, my last slide, because we're also on the orbit 4, this everything has been done with the orbit 4. And now I just gonna change a little bit gears here because we're also interested in protein-lipid interaction in the lab, okay? And we recently find out that this SEC-A, which is in fact a secropin A, it's an antimicrobial peptide in Drosophila. And we found that there is a protein called .A, tron.A, like the opera, the famous opera, I would say, if some people is aware of that. It can protect the host membrane towards the antimicrobial activity. So it protects in a way that is kind of shielding. We have done like some MD on it, shield the PS lipid from the attack of the secropin A. In fact, we use the orbit 16 here to kind of screen different lipid composition, different concentration of secropin, different concentration of TOT-A. And we found out that when we incubate only the secropin, we have a lot of pore formation, as you can see on the top, very nicely. But also when we incubate both the trondot protein and the secropin, you see that the amount of pore is drastically reduced. And we have also some in vitro data showing kind of the same feature. And also in vivo, we did with the Drosophila, we did a KO and overexpression of this, and we observed the same phenomenon. And what is even more amazing is like we also try with the LL37, so it's a kind of human antimicrobial peptide, and it's doing the same effect as the secropine. So we think it's a kind of mammalian eukaryotic effect. And if you want to have a more look, we just recently published this one in bioRxiv. And with this, I will finally go to my acknowledgement slide, sorry. And yeah, I thanked Professor Chankao, which is one of my supervisors, also Professor Matteo Dal Peraro, which is at EPFL, all the PhD students involved in this lab. Soon, Lucien is soon going to be graduated next week, and the people from the LMNN, and of course the funding for giving some money for doing some orbit mini stuff. And Nanyan here, the new organizer, for giving me the opportunity for this talk. Thank you.
Thank you so much for this beautiful talk. Are there any questions from the audience? All right. So I would maybe have like one general short question. So you only consider one artificial membrane with one pore to be like stable conditions for your experiments. If you then use asymmetrical buffer conditions, and you have this pore which is in the ion channel where it's still huge, did you ever calculate if there are any like saturation effects if you have equilibrium after some time that might be a time that's relevant for the experiment?
I think this is something we haven't checked, but it's something that would be interesting to
I didn't do that for alpha-hemolysin, for aerolysin, but I would think it's somewhere in the hour range. So yeah, maybe calculate it for you.
Sure. Yeah.
All right.
Cool.
So that maybe let's thank Juan again.
Thanks.
So that we